Okay, now we've come to our final plenary and our final talk. It's my pleasure to introduce Laura Furlong. She's the professor, she's a, Miguel Servet researcher and head of the Integrative Bioinformatics program of the research program on biomedical informatics in Barcelona. She's the author of many, many peer-reviewed publications. I looked. But we know her pretty well in this community as the author of DisGenet, which is a database of associations between genes and diseases. And she's going to be giving a talk entitled Connecting Genomics and Network Properties of Genes Relevant for Disease and Drug Response.
Excellent. Okay. Thank you, and good afternoon. Let me put it right. Okay, so good afternoon, everybody, and special thanks to the organizing committee for allowing me to present our work here in this conference, and thank you to all of you for staying after this long day. So, as we have been seeing through the presentations of this network biology session, networks are a versatile framework to model the architecture and also the behavior of biological systems. So nodes in the network play particular functions and are important for biological process, and in how they are located in these networks, and I'm in particular focused on protein interaction networks, are related on how evolution constrained their function. So in this context, it's interesting to study the rates of genomic variation and how it relates to network topology to help to bridge the gap between genotype and phenotype. And in our group, we are particularly interested in understanding how genomic variation lead to disease. So in the last years, there has been much research trying to figure out if there are particular network features that can help us to differentiate between non-disease and disease genes. So while cancer genes, and there are a lot of publications on cancer, and here we have only some of them, report that cancer genes are enriched at HAPs in the interactome. For other types of disease genes, the evidences are not so clear. So, for instance, for Mendelian disease genes, they were found to be enriched at HAPs in a literature-curated interactome, but then if you remove from this set of Mendelian genes the ones that are essential, so that are essential for survival of the organisms, they are not longer occupying central positions in the interactome. Other studies reported on the other side that Mendelian and complex disease genes are not so central because they have they are central because they have a high degree and lower clustering coefficients, but other studies using different network properties reported the opposite. So in front of these seemingly contradictory results, we have to think why are they providing these results that seems not conclusive. So there can be several factors. Some factors can be more methodological, such as the source of protein interaction networks or the source of disease genes, or even the actual definition of what we consider a protein interaction network. Are we considering those proteins that physically interact, or are we thinking more flexible definitions such as co-expression or the other kind of genetic interactions and so on? The other problem that has to be taken into account is that we are all aware is the incompleteness of the data we are working on. So we don't know all the genes that are implicated for human diseases, and in the case of the protein interaction network, we know that we have not a complete map about the human interactome, and even for other species as well. Then there is the issue of different, or in some cases discrepant definition of the network properties, and this can be the case of the definition of the hubs. So in some studies, hubs are defined in terms of counting a number of neighbors, but in other studies, hubs are defined according to the distribution of the degree in the whole network. So making the comparison between studies a little bit difficult. And the other reason would be that indeed there is an actual difference in the network properties of disease genes, but when we look at more specific or more different types of diseases. So in front of these discrepancies, we try to approach or carry out a study trying to address some of these points. And the way we did this is as follows. So first, we try to have very clear, or the clearest as possible definition of disease classes, and this is not a simple question, because although there are different classifications and ontologies and control vocabularies that we can use to define different classes of disease genes, it's not easy to find gene sets that are very well validated for different disease classes. So for that, we use DisGenet. So DisGenet has been mentioned previously in other talks. This is a knowledge platform that we develop in our group that provides a comprehensive catalog of human disease genes and also variants, and it can do so because we integrate data from a large variety of resources. Currently, we integrate data from 15 databases, and also we extract the data directly from the literature. As such, it covers the whole spectrum of human diseases, Mendelian diseases, rare disease, complex disease, and so on. So it allows us to extract different sets of genes associated to different broad classes of diseases that you can see here. Complex diseases, Mendelian, cancer genes, and then other classifications based on more clinical taxonomies like the mesh classification. On the other hand, we performed the analysis on a protein interaction network. We took high confidence protein interaction networks from different resources. Actually, we use 8 different networks, and to see how reproducible were the results across those different sources of data. And even 2 of them were derived from single experimental approaches. With regard to the network properties, we propose to use a network analysis that takes into account different perspectives of the network properties, and I will explain in more detail in the next slides how it looks like. And on the other hand, we used information on genomic variation of the genes from large-scale genomic studies to derived gene constraint metrics that allow us to measure the tolerance or the intolerance of the genes to germline deleterious variation. So for us and for many of people that work in network biology, a key point here is to be able to connect the structure of the network with the function. So, and to do so, we propose a multi-scale analysis of the network. So this means to address or to try to measure properties at the local level of the network, mesoscale and global scale. So the local level, popular example is to measure the degree of a node. So are all these properties that measure the direct neighbors or properties of direct neighbors of your node. And another local property would be the clustering coefficient. In the case of the global parameters, network parameters, an example is the betweenness centrality that is based in the calculation of shortest path. Because it measures the distance between any pair of nodes across all the links in the network. And finally, the mesoscale network properties take into account the modular structure of the network. So how the network can be partitioned in modules that are functionally relevant, and thus, if we study how these proteins are connected and located within this modular structure, we can understand how they, in some cases, connect or are important for the connection of nodes within their own module. That would be this case, and we will call this a kind of provincial hub. It has a certain high proportion of edges. And on the other hand, we will have another kind of node that could be important in the network because it's able to connect several modules in the network. So how we compute these mesoscale network properties? So the first part, the first step, is to partition the network into clusters, and for that we use the InfoMap clustering procedure. And after obtaining the clusters, for each node in the network, we compute the two parameters. One is the participation coefficient that measures the fraction of edges of links that the nodes projects to other modules within the network, and the other is the within module degree that is that normalizes the degree of the node compared to the degree of the nodes that belong to the same module. So once we compute these two parameters, the participation coefficient and the within module degree or SID, we can use them as a mesoscale network property, but also we can assign each node according to these parameters what Guimera and Amaral, in a work published in 2005, propose as cartographic roles. So these cartographic roles are depicted here and are obtained by plotting the nodes and in this plane of the within module degree and participation coefficient. So in this case, the nodes that have a within module degree larger than 2.5 are considered hubs, and the other nodes below this value of within module degree and are non-hubs. And then the roles are defined according to the value of the participation coefficient. So we start with nodes with low participation coefficient. That means that all the links are projected within their own cluster. And then we go to the extreme of the scale where nodes with high participation coefficient have all the edges homogeneously distributed among the other clusters of the network. For the nodes that are hubs, Again, we can define the different roles based on their participation coefficient, ranging from nodes that have all the links within their own clusters. These are the provincial hubs, and the kinless hubs that have the edges distributed homogeneously between among the clusters of the network. So these cartographic roles are heuristically defined based on the data. So in the case of the human-protein interaction network, this is how this distribution looked like. So we use this framework to characterize the different classes of disease genes. So in this, here what we show is the results of the over-representation analysis. In this case, we took the whole gene set of disease genes. So this is quite heterogeneous because we put all together different classes of disease genes, and we are talking about around 8,000 genes. And we see that there is an enrichment for roles with high participation coefficient, as you can see here. One of them is also a hub, but the other two ones are not hubs. When we look at the set of genes associated to cancer, we also see enrichment for these high participation roles. But at this stage, and by analyzing the data, we knew that in this set of disease genes, there was a large proportion of cancer genes there. So genes that are associated to Alzheimer or any other disease, but are also annotated to cancer. So we took away these cancer genes from the disease genes and see how the behavior of these parameters look like. And as you can see, all the signal for enrichment for particular roles disappeared when we remove the set of cancer genes from the disease genes, indicating that this signal of centrality was mainly driven by the cancer genes in the disease gene set. We then look at more well-defined or more smaller sets of disease genes. So these are complex disease genes and Mendelian disease genes, where you can see that there is also an enrichment for high participation roles like kinless and kinless hub, so these two roles. And in the case of Mendelian genes, we have kinless and connector roles. But again, when we remove the cancer genes that are also there in these sets, this signal disappears. We then move on and try to look at more specific definitions of disease, and in this case we use the mesh classification. Here we see the upper level of disease classification in which we have like cardiovascular diseases, again, neoplasms, musculoskeletal, and so on. And you can see clearly that there is an enrichment for kinless and kinless hub for most of the genes that belong to the different disease classes. But when you remove the set of cancer genes from these sets, the signal disappears. So we were quite convinced that this reported centrality of genes related to different classes of disease genes were mainly driven by cancer genes in this gene set. So and that we are working with very heterogeneous gene sets. So we then try to work with more homogeneous, if possible, disease gene sets. So for Mendelian disease genes, we divided in autosomal dominant or autosomal recessive, and for cancer, we work with a small set of genes that drive the tumorigenic process, and looking at the enrichment for the cartographic roles, we see that for the autosomal dominant disease gene, The enrichment is observed for kinless and a little bit for connector roles, so again, high participation roles. Again, the signal is also explained to a certain extent by the driver genes within this set. And in the case of autosomal recessive, we don't find any particular enrichment for any role in the network. Okay, so having carried out this network analysis exploring these mesoscale network properties, we thought that these genes that show a centrality at this evaluated at the mesoscale level are constrained or are under parifying selection, and therefore are less tolerant to deleterious germline variation across human populations. And to evaluate this hypothesis, we used data from genomic high-throughput genomic studies aggregated by the ECCB consortium. In particular, we use data from exome sequencing studies, and for that we obtained the variants affecting coding regions and impacting coding regions, like coding stop variants or in-frame variants and missense variants. And we compute the high impact to synonymous ratio as the ratio between the highly impact variants and synonymous variants for each of the genes in the network. And we use this ratio as a proxy of the sensitivity of the genes to likely deleterious germline variants. And when we analyze the different sets of genes with this metric, what we observe are that both the cancer driver and the autosomal dominant genes are more constrained, are more sensitive to likely deleterious germline variants, as you can see here with the set score, while the autosomal recessive genes are are more tolerant to this germline variation. Finally, we were interested to see how these two parameters behave together. So here we plot the set score of the different network features, and here we see the mesoscale network properties, participation coefficient, and within model degree that I presented here, but also the other parameters that I didn't have to present in more detail. And here we plot the set score of the high impact to synonymous ratio. And what we can see is that we obtain a separation between the different classes of disease genes. So in one side, we have the driver genes that have high centrality in the network in terms of both the local and global and also mesoscale network properties, are quite sensitive to deleterious germline variants, while the autosomal recessive gene, Mendelian autosomal recessive genes, do not show centrality as based on the metrics that we use, and are also more tolerant to deleterious variation. So to sum up, we observed that the different types of disease genes have different properties and different patterns of connectivity measured at the mesoscale level. That the cancer genes are responsible for this, for the network centrality of disease genes. And we want to stress the importance of this intermodular connectivity. And that there are different connectivity patterns in Mendelian disease genes, in particular when we look at autosomal dominant and autosomal recessive genes. And also that these genes belonging to different disease classes show different tolerance to likely deleterious germline variation, and that there is a relationship between these centrality measures that we obtain and the sensitivity to deleterious variation. So we then wanted to extend this framework to the analysis of the variability in drugs response. So all of us know that individuals do not respond equally to drug treatments, and we have the problem of lack of efficacy in which the drug does not work properly in some individuals. And the other very important issue is the drug toxicity, when when there is a toxicity caused by the drug. So we also know that there is a genetic contribution to the drug response, and there is a lot of knowledge about variability in drug metabolizing enzymes, and in fact there are many drugs that have pharmacogenomic labeling in their prescription, but there is not so much information on how genomic variability affect other proteins that are important for drug response. And then I'm talking about drug targets, but also other proteins that are important when the drug enters into the body, or also known as off-targets, and also all the proteins and enzymes relevant for drug metabolism. And finally, we also need to understand how the proteins that are relevant for causing the side effects are behaving in terms of genomic variability. So we apply the framework that I explained before to this problem, and for that we compiled 3 sets of genes, one for drug targets, the other one for drugs relevant for metabolism, and this includes metabolism enzymes, but also drug transporters and carriers. And the other set were a set of proteins associated to the adverse effects produced by drugs. And these are the numbers of these sets, and please note that there is certain overlap between them. So for the genomic analysis, we used, again, data from the EXAC consortium, but in this case, we used a gene constraint metric that they provide, that is the PLI, the probability of a gene to be intolerant to loss of function mutations. And in fact, they indicate that these genes with a PLI larger or equal to 0.9 are intolerant, and the other range are more tolerant genes. We also used p-null, another constraint measure, but we will focus only on the PLI. So when we assess the 3 sets of genes for these gene constraint metrics, what we observed is that both the target set and the toxprot set of genes are quite have large values of PLI, so they are quite intolerant to loss of function variation, while the MITAB gene set show a tolerance compared to the other genes in the genome. But you might say that these sets of genes are really heterogeneous, so we look at in more detail on different classes of genes among these large, big sets. So we use the target disease ontology and other other protein classifications to further divide this set of genes. So what we can see is that for the targets, the signal of intolerance to loss of function variation is really high for the kinases, and also for transcription factors and enzymes, and notably there is a class of proteins, target proteins, the GPCRs, that were more tolerant to loss of function variation. In the case of the ToxProt dataset, the signal was more relevant for transcription factor and also for kinases and then these transcription factors. Please note that this set of genes are targets in which we remove the proteins that also produce side effects, and we can see that the signal, although significant, it is a little bit lower. In the case of the METAP gene set, we observed that this signal of tolerance to loss of function mutation was driven by the enzymes within this set. For the network analysis, we apply these different properties, but in this case, we also explored tissue-specific interactomes, basically brain, heart, kidney, and liver interactomes because their relevance to drug toxicity. And to make it short, what we observed is that both targets and ToxProt genes were significantly enriched in kinless hab and kinless roles, while in all the networks tested, and while the METAP genes were not particularly enriched in any role, with the exception of kidney and liver network, in which we found an enrichment for ultraperipheral nodes, roles. And again, when we plot these the network features against the gene constraint metric, we observe the separation between these proteins. The ToxProt proteins are the more that have a high centrality in the network and more intolerant to loss of function mutation, followed by the targets, while the METAP genes that show low centrality measurements and are more tolerant to loss of function mutation. So to sum up this last part, we found that drug targets, and in particular drug targets that mediate side effects, play a role linking different models in the network, in the protein interaction network, and are more intolerant to loss of function mutation than the targets that do not mediate side effects. Within the group of targets, we found that GPCRs are more tolerant to loss of function variation and do not play central roles in the interactome, and the drug metabolizing enzymes are also less central in the interactome and more tolerant to loss of function variation. And to finalize, this is a joint work between different groups. In my group, this was all this analysis was mainly done by Janet Piñero, and Emre Guney, and Kim Aguirre Valdoliva from the structural bioinformatics groups from the GRIP, Abel González Pérez from the Institute for Research in Biomedicine in Barcelona, and Ariel Chernomorets and Ariel Berenstein from Instituto Leloir in Buenos Aires. And with that, I finish, and I thank you for your attention.
Okay, we have time for some questions.
Okay, thank you for your very great talk. So I was really impressed. So my question is, even in the same disease, there are several stages. So I mean the acute inflammation and chronic inflammation. So have you ever tried to identify such different stages by your mesoscale network properties?
This is a very interesting question. We didn't try this, but I think it would be very interesting to see how diseases that are relevant for different stages of disease, if they have different properties in the network, of course. Yeah.
Yes.
How does GeNet compares with ISMB, the Human Gene Mutation Database? What's the difference?
Well, the difference is, as far as I know, IMGD is focused on variants, right? Only
They
mutations.
also have genes.
Also have genes.
Yeah, well, if you have a variant.
I cannot tell you because it's difficult to do a comparison because, as far as I know, it's not possible to download the whole data set in
There is
this.
a version which is public.
Yeah, well, it should be checked. I cannot answer this question. Yeah.
Can I ask two questions? So first question is that whether you look at the cancer of different types, because some cancers might be more heritable than the other cancer types, so that most time you have the somatic mutation, germline mutations more than the somatic mutations in certain subtypes of cancer. So whether you can find a specific pattern for different cancer types. The other question is that when you look at the drug target genes, so can you assume that the drug target genes are a subset of driver genes? Because normally that's how the drug has been developed. They want to target the driver genes. So whether that's the case in your study.
This also are interesting question. I will answer the second first. We didn't assess the cancer genes or driver genes within the drug target sets, but I'm sure they are there. So we could. It would be interesting to see if we separate the cancer genes, what happens with the signal. And for the first question, yes, cancer is really, if you say cancer, you almost don't say anything because we know that there are different types of cancer, and even as one of the talks today, even one single cancer is really a heterogeneous disease. So we didn't check that, but I think that, you can check if you want, if different types of cancer show what are the different parameters. Sure, yeah.
Go ahead.
As your basis network, protein interaction network, you took quite a large number of different networks, which probably have got all different structures. So did you check if there's an influence of these networks, or are your conclusions quite independent?
Yeah, yes, I mentioned, but I didn't go into detail. All the results I show is for one of the networks, but the results were reproducible in all the other networks, but for some very specific parameter that may be not reproducible. Then this was especially for the clustering coefficient that depends a lot on how you count, you derive the edges, and this is important for mass spectrometry analysis in general. Yeah.
Last question.
A very interesting talk. Thank you.
Thank you.
Do you see association between your variables, like different types of variables, and actual overall response to different drugs?
Sorry, can you repeat?
Yeah. So my question is whether the variables that you consider is predictive or show association with actual response rates of different drugs.
We didn't check that, so I cannot answer that.
Okay.
Yeah.
Great. Thank you very much.
And now we have our poster session, but we have 3 poster prizes to give real quickly, and you'll want to stick around because you'll definitely want to visit these 3 posters. We had 4 voters, 4 judges go around. 2 of them looked at every single poster and tried to narrow it down for the second round. The first caveat we have to give is that there are a lot of good